Hello everybody, this is the companion video for Biochemistry 2, Unit 3, Glycolysis. I'm not going to do much explaining in this video. All of the explanations are in the other handwritten, homemade videos. This is just more a quick version of what your notes should look like. So if I'm skipping it over, it's, not that important. It's great for understanding, but this is for, what's on tests right now. It's a good one to know here. This is all you need to know. Coming later. We'll see this in the other video explaining the different pathways that you can do after pyruvate. So this is one illustration. If this helps you, great, we're going to see it again. These next two slides, know them really well, know everything about them. Ideally you would be able to write out the entire reactions, plus the enzymes, plus what enzyme class everything is. Keep in mind you might not see lyase, you might see it's catalyzed by enzyme class 4, for example. Make sure you know all of this again, plus the net reaction. This is the good starting point if you don't know anything else in this unit, if you haven't started it yet. Start here, memorize all of these, then write in the enzymes, then the enzyme class. This is just another illustration. I don't really like this one as much. Same with this. So all of these reactions, I would skip over a lot of the slides. It's not really a priority. If this helps you see, what exactly is happening, that's great. But if you can just write out the metabolites and enzymes, that's most important. Anytime you see energy, it has a magnesium requirement. Make sure you know that. So all your kinases, ATP synthase, they all have a magnesium requirement. That would be the only thing I would look at on this slide. Everything else, don't worry about for now. I talk about this one in the other video, the comparison between the types of hexokinase. Just an illustration. So one question you might see is which one is the heaviest, and you'll see a bunch of metabolites and you might have no idea about them. Remember the first metabolites, they all have 6 carbons in them, 6 carbon molecules, and if you see 2 phosphate groups, remember phosphate is relatively very heavy. So this might be one of the heaviest ones. I'm not going to say it is the heaviest, but if you're talking about metabolites, look for the amount of carbons and the amount of phosphates. Phosphates will be the #1 factor in determining how heavy something is. So if it has 2, it's probably going to be that. He could throw in a trick question and throw in an enzyme. Enzymes are all proteins, therefore they're probably going to be bigger than anything like this. So we're going to skip through all the individual reactions. Make sure you can write them all out. I talked about this in the other video, GAP dehydrogenase. Anytime you see dehydrogenase, you automatically think it's a redox reaction, enzyme class 1, and it has a niacin, it's a niacin dependent, B3 dependent reaction. You eventually will know all of this. I'll take that back. For now, I guess know that NADH transfers electrons, and that's via niacin. Good thing to know about this one is phosphoryl transfer, or substrate level phosphorylation. So it's one of your high energy molecules. We're going to see PEP in a minute here. There's PEP again. So here we go, substrate level phosphorylation or phosphoryl transfer, they're the same thing. They happen in step 7 and step 10. So make sure you know that really well. Per every 1 unit of glucose, each of these steps happen twice. So you get a total of 4 ATP. ATP, sorry, that looks terrible. You might say, this is happening in the cytosol. Trying to think of any other ways he might ask that question. Most of the ATP is going to be produced by oxidative phosphorylation. That's covered in Unit 6. So for now, the only ATP we're really seeing comes from the substrate level phosphorylation. This is a good review, just to know your 3 high-energy compounds. You saw this in Biochem 1. Just another illustration of those. This is the same. He made his own version in slides 9 and 10, I believe it was, where he lists the complete reactions. Definitely don't need to know the numbers or anything. Know that number 7 is done by a kinase, but it is a reversible reaction. What are your irreversible ones? 1, 3, and 10. So you're making ATP. Most of the time, this would be a irreversible reaction, but for some reason, when it happens in the cell, the change in G is not that big. Therefore, it's reversible. Seen this, make sure you know net reaction. I described this in the other video and what enzymes happen, where this stuff happens. So make sure you watch that, understand that well. Another dehydrogenase, nice and dependent. Enzyme class 1. We're skipping through a lot of the stuff in the middle. Is just kind of FYI. I have seen questions about this. What happens when you start starving? To make sure you know you start creating a lot more ketones. Good fuel for the brain. Yeah, those increase. When you're starving, you're running out of glucose, so that goes down. That's good to know. As you start to degrade your fat, and then you start to degrade your muscle, and then you start running out of muscle to degrade. Fat tissue stays pretty consistent. Those are the main questions I've seen asked about starvation. Just make sure you can associate ketones going up with the brain. Oops. Always questions about red blood cells. Always goes to lactate. I illustrate this pathway in the other video, the 2,3-BPG attachment pathway to glycolysis. So make sure you know those enzymes. Anytime you see this, you know it's only going to happen in red blood cells. Let's see many questions on the cancer thing. Interesting to read through. There are some questions about genes, but I don't think it's from this one. I summarized the gene induction regulation in the other video as well. So this is all extra stuff that I wouldn't worry about too much. And we've already talked about the ethanol fermentation in the same looking slide. Make sure you know this is not found in humans. So if you see this whole reaction, pyruvate to acetaldehyde, that can't happen with us. Also in this one, going from, we don't create ethanol, we have alcohol dehydrogenase that works the other way. So if you see ethanol as a product, that won't happen in humans either. But if you see ethanol going to acetaldehyde, that can happen in humans. Again, dehydrogenase, redox reaction. So you can also create lactate in your muscles, vigorous exercise. This is a good slide to know. It's the enzyme I was talking about a minute ago. Alright, so getting into regulation, make sure you know your 1, 3, and 7, your irreversible ones. Know your GLUT families. I've simplified this table in the other video as well. This is the story of the pancreas. Definitely need to know this one. Let's see, pancreas, GLUT2. Here's a better illustration. Understand this one, and this little pathway, be able to draw the whole thing out. Ending with the insulin secretion, leading to PP1. So the different ways it's regulated. I think I have seen this come up on a test. I would know that muscles last the longest. Liver has a quick protein turnover rate. I would know your 3 amino acids that have this OH group attached. That OH group is a good binding site for the phosphate group, which turns things on or off depending on the enzyme. So just kind of giving the general overview. Again, all of this is explained in the other video. Make sure you know the full reaction. Don't worry about that. I referenced the AMP kinase slide. Be vaguely familiar with the effects of AMP kinase. It's a great summary slide. This one again, don't worry about that. Liver sequestration. Make sure you can explain this. You're going to see glucokinase. It's much more likely than hexokinase 4. This is asked quite frequently, knowing glucokinase is a glucose sensor. So make sure you know how these parts of the regulation work. So this is in muscle. This is how you turn it on in muscle. And it's hard to say which slides to look at and which ones not to look at for this 2,3-BPG, PFK1, because there's so many of them. I definitely want to look at that one. Another pretty heavy molecule. It's got 2 phosphate groups in it. It's going to activate PFK1. Make sure you know that. This slide really does a good job of explaining it. I think it's helpful in explaining how this part works, especially in the middle here. So if you understand this slide, you really understand the glycolysis regulation, the hardest part of it, really well. So I'll spend some time looking through everything on here. It's going to come up again in gluconeogenesis, Unit 9 or Unit 8. So just learn it now. So reaction 10 is also regulated in a similar way, except less complicated. So that's a good one to know. Kind of more overview material, talking again about the The energy charge is mainly associated with muscle. The liver is a little more complex, associating with fed state or fasting state. This is the gene thing. I think that's all you really need to know. That's what's in red. So don't worry about the other slide on gene induction as much. This is summarizing a lot of what happens when you have insulin as far as regulation goes. So if you understand it all, you should be able to kind of summarize this yourself. This is another summary slide. It's, I think, just kind of simplifying a lot of this here, and remembering that Unit 2 insulin pathway can come up, and he might combine Unit 2 and 3 questions. So when you eat, what's going to happen? The whole insulin cascade, GLUT4 is upregulated to the cell membrane. PFK2 turns on, which makes this here, which turns on PFK1, and glycolysis happens. You create more ATP. So basically the opposite happens for the most part. When you have glucagon, you're not going through glycolysis because pKa is active. It's another good one to look at that summarizes all the regulation stuff we've talked about already. Same thing with this, remembering that pathway with glucagon and cyclic AMP from Unit 2, and then applying it to what we just learned in Unit 3.